Welcome to the course on resistance of arthropods to pesticides. In this course, myself, Marie-Stéphane Tixier, and Anastasia Tsakaraku will present different aspects. What are insecticides? Insecticides are molecules that kill insects, and they can do it in different ways. They are small, organic lipophilic molecules that can kill insects because they will interfere with nervous system, growth development, or respiration. Most of insecticides act on nervous and muscular systems. They usually act rapidly, causing convulsion or paralysis. Growth regulator insecticides target cuticle formation or lipid biosynthesis. They act by mimicking growth or mold hormones. Several insecticides are known to interfere with mitochondrial respiration by the inhibition of electron transport or oxidative phosphorylation. Some insecticides, especially microbials, act as disruptor of mid-gut membranes. Finally, you should know that for some insecticides the targets remain unknown. The table provided by the IRAC, Insecticide Resistant Action Committee, provides a synthesis of all these modes of action. Knowing the mode of action of an insecticide is indeed a key issue to manage resistance. Now, what about the context and the development of resistance? First, it's important to know that insecticides, by efficiently killing pests, are useful for producers. Their use has contributed to the increase in the 20th century of the agricultural productivity and to the control of some human diseases as malaria. However, the increased use of insecticides, besides to lead to environmental pollution, also led to the development of resistance in the targeted organism. In USA, the economic cost of resistance has been estimated to $40 million. How resistance can be defined? On this slide, you can see the IRAC definition of resistance: an irritable change in the sensitivity of a pest population that is reflected in the repeated failure of a product to achieve the expected level of control when used according to the label recommendation to that pest species. In other words, when the recommended dose is applied several times, the product doesn't kill the target anymore. Among the species with recorded resistance to many different pesticides, we find spider mites, and insect pests, and virus vectors such as waterflies, aphids, and thrips, pests that are studied in the European project. We are going to see now how the resistance develop. Insecticide resistance is an evolutionary process resulting from genetic adaptation of pest population to pesticide applications. Within a population, it exists a very low density of specimens bearing a pre-existing mutation that allows the insect to survive after the insecticide application. More insect density is high, that is to say, more its fecundity is high, higher will be the number of its particular genotypes within the population. If the mutation present in this rare specimen provides a biological advantage, these mutants will be selected. It's what happens with insecticide resistance. The application of the insecticides acts as a selection pressure. When an insecticide is applied, it will kill the majority of the specimen, but some will be able to survive. Several ways for an insect to resist to insecticides, namely behavioral resistance, resistance due to a reduced penetration of the insecticide, resistance due to the modification of the target, resistance due to the destruction of the insecticides, and finally, resistance due to the insecticide sequestration. We are going to see and explain all these mechanisms. Behavioral resistance occurs when insects avoid contact with insecticides. Two cases can be distinguished. In the first case, the insect recognizes the applied molecule as a danger and escape. This behavior has been essentially observed for Diptera. In the second case, the insect does not move to avoid the contamination when walking on the leaf. This behavior has been observed for larvae of Lepidoptera noctuidae. Structural changes in the integument of the insect may affect insecticide penetration. It's a physiological resistance that is related to the kinetic of penetration of the insecticides within the insect body through its cuticle. In resistant insects, the molecule penetrates more slowly and or might be destroyed or excreted. This kind of resistance depends on the nature of the insecticides and of the cuticle. It should be known that this mechanism has been little studied. An important resistant mechanism is the detoxification of the insecticide by enzymes that transform the insecticides into non-toxic compounds. Once in the body of the insecticide is degraded by a variety of enzymes. The main enzyme families are the monooxygenase cytochrome P450, the esterase, and the glutathione N-transferase. Resistant insects degrade the insecticides faster than susceptible ones because they possess more effective forms of enzymes with a higher catalytic rate or because the enzymes are produced in higher quantities. Detoxification can occur through degradation or sequestration of the insecticide molecules. Enzymes can detoxify xenobiotics by changing their molecular structure into non-toxic compounds, as we saw previously. Enzymes can also transform the insecticides into stable compounds that are no longer available for chemical reactions. They are suitable for a rapid elimination from the body, and this is a sequestration mechanism. Another major resistant mechanism is a modification of the targets. Target resistance is the second most common mechanism after detoxification. It's also called altered target site resistance. In the susceptible insect, the insecticides reach the target and bite on it, leading to the inhibition of the target function. The insect cannot survive and die. In the resistant insect, the shape of the target has changed, but the target is still functional. The insecticide cannot bite the modified target, and the insecticide is thus not active anymore. It's why the insects do not die. This resistance is due to mutation in the insecticide binding site of the molecular target. This mutation reduces target affinity with the insecticide.
As we already discussed, under continuous selection pressure, resistant insects outnumber susceptible ones, and the insecticide is no longer effective. We showed that the speed with which resistance develops depends on several factors, including how fast the pests reproduce, their migration, the availability of nearby susceptible populations, and the rate and number of applications made. Resistance increases faster in situations such as greenhouses, where insects or mites reproduce quickly. There is little or no immigration of susceptible individuals, and the user may spray frequently. It is extremely important to take action in order to delay resistance or to manage it in case that it appears. Respect the recommended dose, alter groups of mode of action based on scientific knowledge. Reduce selection pressure by diversifying control strategies and apply integrated pest management. The availability of tools for the early detection of resistance and for continuous monitoring is also of high importance. Equally important are decision-supporting tools such as databases where data on resistance is systematically stored and easily retrieved.